So now let's welcome Mitya for the first lecture.
The notes view. Yeah, perfect. Oh, perfect. Good morning, everyone. So I've been told that my last name is difficult to pronounce. So I made a little pictogram here. This is from Austin Powers. That will help you to know how to pronounce my last name. Okay. So it was great to have this introduction. I think that, we all want to understand how the brain works. But just to sharpen this topic, I want to start with a question. So why aren't we there yet? Why can't we model the brain the way we can model physical systems? What I mean by that is, why can't you model the diagram that Rava put up, where given sensor inputs, you can predict what an organism would do in response to that? What is the behavior? So does anyone have any ideas? Why aren't we there yet? What is the main bottleneck? What is preventing us from doing that? Excellent, excellent. So can I just summarize it? Biological complexity. Yes? Any other thoughts? Okay. You don't know all the relevant parameters at the same time. Any other thoughts? Excellent, excellent. So multiple levels of complexity. Excellent. Biological systems are not deterministic. Right. So we can combine this together with non-deterministic, yes? Okay, excellent. Anything else? So it's hard to characterize and control inputs and measure outputs. Okay, excellent. Yeah, that often happens to me, yes, I know. Right, it's not a static system, it's a dynamic system constantly changing. Okay. Right, we don't have a mathematical abstraction to incorporate all the data, okay? Any more thoughts? Okay. All right, so this is all great, and this is absolutely a big problem. And so I actually wanted to, simplify this a little bit so we don't work on our brain, work on this C. elegans brain that has about 300 neurons, and for which the connectome is known, and even the synaptic weights are kind of known, and sometimes even the signs of the synapses, but we still cannot model it. And so some of the arguments that you made apply here, and what can we do about it? And one approach that I'm going to talk about today is to try to complement the lack of experimental measurements of all the parameters in the system, which there are many even here, by hypothesizing about the function. And this is what's called the normative approach. So, normally in scientific method, we go from experimental data to theoretical model to predictions, and then we close the loop, hopefully. But if there is not enough experimental data, like someone said, okay, we can't know all the parameters of all the neurons and all the synapses at the same time, and we can't always combine results from different animals because they're not the same. So our model, our model has a lot of uncertainty, and then we cannot make good predictions. And the idea of the normative approach is try to help that situation by saying, okay, whatever the brain does, it has been optimized by evolution to do it optimally, right? Because, we come from the long line of ancestors, and they were really good at maximizing fitness and so on. And so we can hypothesize that there is something, the mechanisms and the models that describe those mechanisms have to be optimal on some kind of tasks, okay? Of course, as Rava was saying, you can apply this approach in different levels. We will apply this on the very mechanistic level of individual neurons and synapses, but you will see how that works in practice. So this is kind of abstract. So if there are no more questions, I'll go on to the specific example, okay? All right. Yeah, I'll figure out, if I figure out how this works, okay. So, so the key step in this normative approach is to figure out what is actually being optimized. And there is no other way but just to guess it. Of course, it can be an informed guess. You look at the data and you form a guess, and then you pursue that guess and you see where it leads you. So in the case of a neuron, if you want to ask a question, what does a neuron do? So we kind of know that, it can get thousands or tens of thousands of inputs, and it sums them up with certain weights that are determined by the synapses, right, that those inputs go through. But after the summation, the output is a scalar, a single number at any given time point. And that scalar is being sent out to thousands or tens of thousands of downstream neurons, okay? So you can see already that both anatomically and physiologically, it's very, very salient that a neuron is reducing the dimensionality of the inputs. That the inputs could be 1,000-dimensional, 10,000-dimensional, but then that huge vector, that very high-dimensional vector, gets projected onto a single number, which can be like the firing rate or the membrane potential, whatever you like. We will not worry about spike timing in this talk, so just think about graded neurons or firing rate neurons. Okay, so that huge, that high-dimensional vector is projected in one number. So that's dimensionality reduction. And then you can figure out this. Okay. So we can write it down in a mathematical form. So are you okay with this notation? So the bold-faced letters will be vectors, and this is transpose. So this is an inner product. Is everyone okay with that? Okay. And then we require that the norm of W is 1. Okay. So that we assume that the sum of the squares of the synaptic weights is 1, and then y is a projection of this vector x onto w. That's basically what a neuron does. We know this from experiment, and we can also show this geometrically. So we cannot plot 10,000 dimensional space, but at least for 2 inputs, we can plot the data on the slide, and this is, say, 1 data point at time t1, and then there is other data points that came in different times, and then there is some vector w on which the data gets projected, okay? And that's what the synapses are doing, okay? So that's like a view of what a neuron does. I'm just retelling you what you already know based on the summing of synaptic currents, okay? So, but now we will ask a question, well, but what determines that vector w, the synaptic weight vector, on which the projection of the inputs takes place? And that's where the normative approach comes in. The hypothesis is that when we reduce the dimensionality of the inputs, we kind of compress the data, but we want to preserve as much information as possible. And the way we will quantify the quality of our compression, the preservation of information, we will minimize the reconstruction error squared, which is what we do is we say, okay, suppose there is this virtual step, which is now called decoding, where we say, okay, someone told us that we now, based on y, we need to reconstruct this vector of inputs xt. And our reconstruction is called xt hat. But we have to produce it just from y. Okay, it's intuitive that all we have to do is scale w by y. And then what we need to do is we need to minimize the error between the reconstructed xt hat and the actual data. Okay? We're okay so far? So what this means, for example, this is xt hat, and this is xt, and this is the error for this data point, and then there are errors for other data points to minimize the total sum of errors squared of those points. So that's the guess for our normative approach, and we are going to take this guess, do the math, and see what it predicts for the synaptic weight vector. Any questions? Exactly. So for a neuron, there are like thousands of upstream neurons, and they generate spikes, and we quantify those spikes by firing rate. So we assume that at every time point, there is a vector of firing rates impinging on that neuron. And that's what the vector xt is. Does that answer your question? Right. So if all, you're correct. So if all axes were pointing in the same direction, then it will be very easy. That's the direction of W. But at every time point, there is going to be a different combination of firing rates on the upstream neurons, right? Just thinking about like recording from the brain from 1,000 neurons at a time, they all have different firing rates, and those firing rates change over time. No, there is there is a vector W, which is this bold letter, which is only one per neuron, okay? And then the component of each vector is like W1, that's what multiplies X1, W2 multiplies X2. But the problem is that there is also a time dimension, and so at every time you get a new vector X, and you have to project it on W, to compute y. Exactly, exactly. But it cannot, W is trying to please, but it can't please everyone. If it pleases this x, it will not please that x. So it's trying to find the golden middle, right? Okay. There was another question, I think. Uh-huh. So it's a guess that the job of a neuron is to project this high-dimensional vector x onto a single dimension, y, so that from that y, we can virtually reconstruct x back by multiplying it by w. Okay? Of course, no one in the brain is going to do the reconstruction. There is no homunculus who sits there and reconstructs the world, right? That's not the goal. The goal is to generate behavior out of this, okay? But you can't generate behavior if you don't have the information about the world. So you need to preserve the information in your observations to generate behavior. And in the simplest possible way, we can say, okay, well, we want to make the y that is most informative about x. Does that help? Exactly, exactly. So it's when it's bold, it's a vector. When it's not bold, it's just a number. Okay?
Each neuron presumably is doing something different, and if you wanted to just preserve information, the best thing would just be not to have that neuron and just to use the inputs directly. So,
yeah, okay, so very good. So
shouldn't you think of what the postsynaptic neuron,
exactly.
what that function,
Exactly. So that's an excellent question. So the question is, why not just like deal with the data that we get it? And this is like, you know, why do we do linear regression, right? You're in a physics lab, you do a lot of measurements, right? But you can't just use those measurements to say something about the system. You need to abstract the data to extract some kind of law that will allow you to make predictions about the future so that they're generalizable. So in physics, for example, or in statistics, what you would do if you have a data set, you would compute linear regression, and that would be the best prediction for another data point. When another data point comes in, it will lie close to that line. So that's the idea here, to come up with a description of the data that is generalizable to other situations. And so you can't just like keep all the data points. You really have to abstract them to a lower dimensional space to make a prediction. Okay? Does that help?
ability. So maybe you should, in what way? Maybe you can expand on this.
Okay, let's. You're jumping ahead. Okay, so ask me this again in a little bit. Okay, so the reason I'm kind of like cagey about this, I'm assuming that the data comes from a distribution which is characterized by a constant W. So long as that W that generates the data is constant, then we can do this. Okay, then it will generalize. But if W changes in the world, then we'll have trouble with generalizability. Okay? Is that okay? Are there other questions?
Can we reconstruct the signal to another direction?
Yeah, so that's a good question. So this is kind of intuitive that if you use W for a projection, then WY is the best estimate of X. You can do this. You can actually show this if you just say, okay, let's move on a little bit. Okay, so here W transpose X comes from here. That's encoding, and then W, this W is decoding. Right, so you can actually have both of those variables different and optimize with respect to those, and then you find that the optimum is when they're the same. And that's kind of intuitively clear. If that's what you use for projection, that's the best thing to reconstruct. Okay? Any other questions? All right. So now we have this approach. So we have an optimization problem with respect to W and Y. We can plug in Y and we get expression just in terms of W. And then when we expand the square, we find that there are two terms that depend on W. And the two terms actually are the same because the norm of W is 1. And instead of minimizing the error, we actually can maximize this kind of expression, which may look unfamiliar, but it is actually something you very well know, right? Because we can move the sum inside and sum over the data, and this is just becomes what statisticians would call a sample covariance, right? This is just the covariance matrix of vector X, and then written this way, this is like a Rayleigh quotient. So it gets maximized by the top eigenvector of the covariance matrix. Okay? And this is something that you probably heard about in statistics. Do you know what this method is called? So you find a direction of the data that maximizes the variance. PCA. That's what this is. So the interesting thing is that PCA is usually formulated this way, is maximizing the covariance, the direction in which the projection has the most, the direction along which the projection has most variance, this is, but it's actually equivalent to minimizing the reconstruction error. So there are two equivalent definitions of PCA, and we started with one, which was justified, motivated by the normative approach, by the minimizing of the compression error, and got to the other one. But we are in familiar territory now because what I'm telling you is that this normative approach suggests that the neuron is doing PCA. You might have heard about this from a different direction. This is also what's called autoencoder in machine learning, right? So you take the data, you compress it to one dimension so that the virtual reconstruction is as close to the original data as possible. And as you know, autoencoder plays an important role in modern AI. You know, inspired eventually things like ChatGPT. Okay? All right. So the prediction is then from the normative approach that a neuron does principal component analysis of the data. Now, of course, this is kind of abstract, and we assumed a lot of simplifications about the neurons, ignored spiking, and so on. So does it have anything to do with biology? And so we decided to test this prediction, and the prediction is that the synaptic weight vector will be the top eigenvector of data covariance, so that would be the principal component. And we were able to test it in a particularly simple neural circuit, or relatively simple neural circuit, the olfactory system of a Drosophila larva. So before Drosophila becomes an adult and flies away, it's like a little worm larva, and its olfactory system has just 20 neurons, each of which uses a different olfactory receptor. So each of them responds differently to the odors. And the circuit that is downstream of those receptor neurons has been completely mapped out. There is a full connectome. And one feature of it that we'll pay attention to is that there are this inhibitory interneuron, like this one, and they provide feedback inhibition. And also from connectomics, we know the synaptic weight vector of these connections from those 20 neurons to this interneuron. So there's a 20-dimensional vector of synaptic weights. And in the same system, another lab has measured the activity of those ORNs. So these are different ORNs. They all have names according to the receptors that they express. And this diagram shows the response measured with calcium indicators of those olfactory receptor neurons to different orders. So there is a bank of orders that have been presented to the larva. And in response to each order, there is an activity in those ORNs. And because different ORNs express different receptors, their activity vector is different for different orders. And that's how the larva and actually all the animals can tell different smells. Okay? But now we have this data of all the neurons responding to all the odors. And this is just like having those vectors X, right? So different index T corresponds to different orders. Different components of X are different ORNs. And so this is a matrix of activity on which we can compute the principal component analysis. And so we computed this principal component analysis and compared it to the synaptic weight vector from those neurons to the interneuron.
Where is the data on I in your...
So I is this postsynaptic neuron?
Yes, but you don't have data on that.
No, I don't.
So how are you going to reconstruct the synaptic weight of I?
Oh, the synaptic weights were measured anatomically from connectomics. So what happens in the fly is that all synaptic contacts are approximately of the same size. And so you can just count the number of contacts that two neurons have, and that's a good approximation of synaptic weights.
Yeah,
So
but data that you're going to compare, I you have not recorded, so how are you going to compare with I?
okay, so I'm just comparing the synaptic weights that were measured anatomically with the PCA of the activity vectors on those ORNs.
Yeah, but these ORN are presynaptic to W, so,
Exactly.
so they don't give you any indication on W.
No, they don't, but I'm predicting, right? So we're predicting that this synaptic weight vector finds the direction that corresponds to the top eigenvector of the covariance, right? So I compute the covariance of the data from calcium imaging and compare that vector with the synaptic weight vector. Is that okay? Okay.
Synaptic weight you have from connectomics.
Synaptic weight vector comes from connectomics. Okay, so that's this W, and we compare that with the direction of maximum variance in the activity data upstream of that neuron. Right. So, okay. So the question is there a single postsynaptic inhibitor neuron multiple? So the truth is there are actually multiple, and there are 3 of them for which the synaptic weight vector is approximately the same, and they kind of all work together. But there are a couple of others that we are ignoring here. So we're just taking the one that that has the most influence, we think. And that one, in that one, we can explain synaptic weights using PCA. For the other ones, we cannot. So that's the limitation. We cannot account for multiple synaptic weight vectors. We can only account for one.
I mean, the difference you are mentioning, like these eyes having like a weight, like a PCA versus not, like it might be related to the feedback inhibition it's sending.
Yeah, so there are a lot more complications, and the actual diagram is more complicated. But this is the main kind of part in terms of synaptic weights. This is the central part. So of course, we can't explain everything at this point, but we can capture the synaptic weight vector in this projection onto those principal interneurons. Yes. 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 So the theoretical, his name appears in the next slide. Yeah. So this was proposed first by Oya. The PCA neuron was proposed by Oya, but it has never been really tested because we didn't have the data. And so now we have the data, and there is at least one case where this seems to agree with the experiment. Yeah, so, okay, there are a lot of holes in this experiment, right? Or rather in what we did. The experiment is good. So I'll explain to you how these orders were chosen. So these orders were chosen so that they're as close as you can come to a ligand, which means that they found orders that stimulate one neuron the most while leaving other neurons inactive, right? So this is the favorite orders for each one of them. And so the idea why that is a good, the good test set is because it kind of explores the very kind of corners of this high-dimensional olfactory space. Of course, this doesn't mean that it captures the statistics of the previously seen orders, which is what you really need. This is the expectation value, right, in statistics. So we of course, we're not able to get that data because we don't know what larva explored. But we try to just get as far as we could with the data we had, which is the most diverse set of orders already you can use to compute the PCA, and it gives you the synaptic weight vector. Well, we would, yes. Yeah.
Activity or something instead of finding the PC.
Yeah, so that actually goes back maybe to Rava's question is, what is it that you want to do with the principal component that is in the order input? And in this case, it has a feedback inhibition, means that the brain actually wants to discard that information. So as you can think of PCA as finding the most correlated direction, and so you want to get rid of those correlations because they're not very informative in this case, right? So you're trying to decorrelate the data or whiten the data to get rid of the stuff that is always there, right? And this has to do with the fact that in biology, unlike physics, you can't always have an orthogonal basis for your measurement, right? So the way these different ORNs respond to orders is that they don't span the space orthogonal, right? And so there are correlations that are built in their responses, and they're always there. And that's why you want to get rid of them. Okay? Any other questions?
And then
you see which neuron fits this model, or do you have to study this neuron and come up
So,
with, oh, maybe this is a PC?
Okay, this is a good question. So we did not have this data when we came up with the model. Well, Oya came up with the model, okay, and we did something else that you will hopefully hear about in the second part of the talk. But then when we heard about the existence of this data, we decided to test it, and we found that there are neurons there that fit this model, and we did statistical analysis in the paper. Nikolai, who's the first author, he did statistical analysis very carefully, trying to make sure it's not just a coincidence, but it is a real statistical significant result. Okay? Any other questions? Does it suggest what?
But it suggests that the neurons act like linear system.
Okay. So that's a very good question. So the assumption that we made, of course, that neurons act as linear summators, but we know they have a nonlinearity. And so actually the second part of my talk deals with the nonlinearity. And this is just a starting point where we want to, it's like in physics, we want to solve the simple problem first.
Okay.
And this is just a sanity check that we are on the right track, that it's not too far off.
Okay,
but
thank you.
yes, there are a lot of limitations to this model.
Okay, thank you.
Very good question. Any other questions? I mean, it's up to the students. I can.
You want to have a small break in between? Everyone is okay? Okay, keep going.
All right. Okay. Okay. So good. So at least we are not completely off. So now let's go back to what this normative approach framework does for you. So we started with this guessing the objective function, minimizing the reconstruction error, and then we said, okay, we can do this by projecting the data onto the synaptic weight vector, and that's what neurons kind of do. And then we have a model of a neuron that does it, a linear model at this point, but we do have a model, and so that's all good. But there were already questions asking, wait, but where will you come from, right? How does the neuron or the olfactory system of the larva know what the principal component of the order ensemble is? And of course, this could be influenced, set up by the genes to some extent, but there is now data actually from a lab here in Beijing that W can be a function of the exact order background that the larva is brought up with. So it's learned. So how can W be learned from the data? And you say, okay, it's very easy. Just take the top eigenvector of the of the data covariance. But how can a neuron find the top eigenvector? So how can a neuron do PCA? And that's what Oya figured out in 1982. He basically said, okay, if you have this learning rule where you adjust the synaptic weight based on the reconstruction error times y times the output of the neuron, and eta is the learning rate, then W will be the top eigenvector of variance. Where does this come from? Well, the derivative of this objective with respect to W, and then from, this is like xt minus wy transpose times x minus wy. So from the first bracket you will have y, and then you will have the second bracket. That's the gradient descent. Okay? So the gradient descent on this objective function just gives you Oya rule. Of course, the neuron cannot compute the gradient descent for the whole data set, it has to compute it online because the data is streamed, and it has to compute the output y in real time. Otherwise, you know, what's the point? And so there has to be an online updating rule, and that's why that's what this OIA rule accomplishes. Okay. So graphical terms, the data comes in, and the W gets adjusted, and when every data point comes in, as someone correctly noticed, W won't just represent that data point, so it gets pulled in the direction of each data point, but they all pull in different directions. So in the end, the W finds the direction corresponding to the top eigenvector of the data covariance. That's the result of this tug of war between neurons pulling W in different directions. Question? Why is there YT? Sorry, I didn't understand the question. Say it again. Oh, so this, the bracket, you're okay with the bracket. This is the error. Okay. But you don't want to listen to every data point equally. You want to weigh the contribution of data points to W by Y. So that's the difference between linear regression and PCA. Right? So in linear regression, you just minimize the error. In PCA, you minimize the error weighted by the projection of that data point onto the W. Because if the data point is orthogonal to W, you don't want to represent it. Another question? So I'm not sure I completely understand the question. There is no decoding step in the brain. So the way that the synapse knows the error is because it gets Y from backpropagation, right? So there is backpropagation when the spike is generated by an axonal hillock. There is a propagating signal down the dendrite back to the synapses. So and that's like there is a calcium and so on, and the neuron, the synapse knows that there was a spike produced by that neuron. And that's how it has access to Y. Of course, it knows its own synaptic weight, and it knew the component of X that came in. So that's why it knew everything. The synapse knows everything it needs to know to do this update. Inside the neuron, there is a feedback. But there you don't need error backpropagation like in machine learning. You don't need that. Everything is local. Okay. Yes, thank you. You I'm guessing my next slide. Yes. No, it's good. Of course, the reason we're sticking with this rule is because there is experimental data that supports that synaptic weight is proportional to the correlation between the pre and postsynaptic activity, which is what this main term is. Okay? So this is the Hebbian plasticity in OIA's rendition that is an experimental, provides experimental support for this kind of framework. So there was some other question that I kept, yeah, yes. Oh, okay, okay, okay. Is it what? Yes. Yes. So basically what's done here is that this is the objective we want to optimize, but we have to optimize it online. So at every time point, the data vector xt comes in, gets projected onto W that was there before, and that's yt. And then after that, W is updated based on this reconstruction error that is computed by backpropagation, and then there is a new W like I was trying to show this in this animation. Okay, so that every time the W gets updated, and there is a new projection. Okay, so that's what's going on. Where is what? Initial condition? You can start from a random from a random initial condition. Okay. The question is, if you have a lot of data, eventually it will wipe away all the previous memory that was in the initial condition. Ah, okay, excellent. Oh, very good. So you guys are paying attention. So my answer to the previous question wasn't completely correct. So you can start with a random initial condition, except for the set of measure 0, which is the orthogonal W to X. If that's the case, then you will never get a non-zero Y, and then this will never get off the ground. Thank you. Always good to have a mathematical rigor. So other than that, if I just randomly choose and I don't hit the orthogonal vector, then I'll be okay. Okay? All right. So yeah, so it's very important to mention that this kind of learning rule is local. That means that a synapse has access to all the numbers that it needs to for the update. Yeah. So I think the question was, if all the data comes from the subspace that is orthogonal to the W, then Y will never be non-zero. If Y is never non-zero, then W will always stay orthogonal to the subspace of the data. Okay? But that's, okay, we can assume that that's not going to happen. Okay? All right. So yeah, so it's very important that this is a local Hebbian learning rule, and therefore it can be implemented in the brain with biological hardware. So just a question for you guys. Do you know of a non-local learning rule? What is an example of a non-local learning rule? Right. Error backpropagation used in machine learning. So it has no chance to be implemented in the brain because there is no physical substrate to do that. Okay. But this has. Okay. So now this is what the kind of the maybe the simplest case of the normative approach applied on the levels of neurons. Start with optimization. Okay, I did this again. Started with optimization problem, derived an online algorithm, and compute a neuron model. Before we go on, I just want to give you yet another visualization of what the neuron is doing. In this case, using the language of linear algebra, and in this case, we're solving the same kind of problem, but we will rewrite it using the matrix notation. So we'll take all the data vectors xt that came in different times and stack them together to form a big matrix X. And then you can write this objective as a rank 1 factorization of x, where x is basically written as a product of the weight vector times the raw vector, which is yt. Okay? So that's just PCA, and that's the same thing you know about. I just want to show that how it works in real time. You get more data with time, and you get more outputs, and the W changes with time, and that's what the flickering is supposed to indicate, right? So the vector W gets adjusted, and the output of the neuron gets produced as you get the data. So the data is streamed in, the output of a neuron is streamed out, the W gets adjusted, and the life goes on. So that's the life of a neuron according to this model. Okay? Well, this may be good, and we may be, you know, have some experimental consistency, but I don't think we'll get very far in life if we just use a top principal component, right? Unless our world was one-dimensional, a top principal component won't get you very far. And you saw the olfactory system, we're just going to, you know, it was just getting subtracted. So we want to look for more principal components, for more features. And so for that, of course, we can't use one neuron, we have to use multiple neurons, because an output of each neuron is scalar, so it can only capture one principal component. So to have two principal components, you need two neurons, three neurons, and so on. Can we do that? Well, so that's why I showed you this matrix factorization visualization. So the data is still this big matrix X, and now it has to be decomposed into higher rank factorization. And you can think of that factorization as a sum of rank 1 layers, right? So matrix can be reconstructed as a product of these matrices or a sum of this rank 1 products. And then the idea is that for each such rank 1 product, you would use a separate neuron. And then if you had two neurons, you can have a rank 2 approximation to this data matrix. You have 3 neurons, you have a rank 3 approximation. Excellent question. That's exactly what I'm going to say next. Yes. So first of all, how do we even learn this principal component analysis? And you say, oh, yeah, you know, the Euler rule, right? You just told us. So why doesn't each of those neurons run an Euler rule? But that's not going to work. Does anyone know why? Exactly. So because there is no interaction between neurons, each neuron will learn the top component. So what are we going to do? There needs to be some kind of communication between those neurons. They're telling each other, all right, I'm covering this top component, so you don't have to. Okay? So there needs to be some kind of lateral connections that tell other neurons this component is taken care of, presumably inhibitory, right? So, but now there are a lot of connections, and what are we going to do? How can we build a network that really computes this PCA for multi-channel case? And we go back to our favorite normative approach, right? Now we write down the objective in terms of this matrices, this big data matrix X, and now this matrix W, because there are multiple neurons, so each of them has a vector of synaptic weights, and so together they're matrix, and this is the output matrix because there are multiple channels. And we take a derivative like we did for the Euler rule. First, we take a derivative with respect to y. Sorry, I shouldn't have said that. First, we take a derivative with respect to activity on each step, and we take a gradient with respect to y. And if you take a gradient with respect to y, you get this expression. And these W's are matrices, and they multiply these vectors. And this can be done in this very biologically plausible neuronal circuits, right? The first term are these feed-forward projections, where the elements of this matrix W are synaptic weights in those connections. They multiply the input vector X. But then we have the second term, which came from the quadratic term if you were to expand the square. The quadratic and y term if you were to expand the square. And these are those red connections, the lateral connections. The weight of those connections is this given by the elements of this Gramian matrix, which is the product of two matrices. That sounds a little bit complicated, but okay, anyways, with just synaptic weights, we can do that, right? So this kind of network will compute Y that would optimize this framework. And this is how we can get different principal components. Of course, I have to tell you what the learning rule is. And the learning rule we can obtain by taking the derivative of this same objective with respect to W, just like Oya did. And so then we get eta, y, x, all good. But now we have to sum over all other channels. And this is a big problem. This is why there was a question that if you were to do this, the learning rules will not be local, right? Because the reason I kept all the subscripts here, so the synaptic weights for a synapse, say, from input 1 to output 1, depends on the activity of the output 1, y1, the activity of input 1, x1. This is all good because the synapse has access to those activities. But this term is horrible because it requires the synapse to know the activities of all other neurons weighted by the different feed-forward weights to update itself. And how can the synapse possibly know that? So that's why this rule is non-local and cannot be implemented biologically. Right, so this is a good question. So why do I insist that the rules are local? Because there are physical substrates that can communicate among all the neurons non-locally. And that's a fair point, but the problem is that these substrates, there are very few of them. And so you can only have a few global signals. In this case, it's not going to work because each synapse will require different combinations of those inputs to know about what it needs to do. So if there was just one general global factor, I think that would be okay, that would be biologically plausible. But here you can see that this W weight depends on the sum, which has an index J in it. So it's specific for each synapse. And so for each synapse, you have to recompute a different sum. And that would be difficult to do because then you would have to have an astrocyte for each synapse. And then how do you tell those astrocytes what to do? Then they have to have a learning rule of their own. And then they have to, each astrocyte have a family of its own astrocytes, and then it goes crazy, okay? So that's why, yes, you can have a few global factors, but not enough to specify the learning in each synapse. Okay? Yes? Yes. Okay, well, I'm not sure I completely follow your question. So, so what we are doing, like stepping back, is trying to understand the computations that the neurons are performing, right? And so that computation may be implemented differently if you had different hardware, right? So if I had a laptop, I can compute PCA by just running it on a CPU architecture by taking the top eigenvector. I don't need all this, right? But biology cannot do this. Okay, now if you're asking, well, why to be bothered by biology if we can have ChatGPT, the idea is that, well, ChatGPT can't do everything humans can do, so there must be some kind of a secret sauce here that we are trying to discover. So it will turn out, actually, I believe, that these constraints that seem to like handicap you and make you like to fight with one arm behind your back, right, are actually going to be helpful. They're like guardrails that point you in the right direction. But then it just gets one top principal component, but there is more information in the data. Sorry. Okay. So there is more information in the data than one principal component because it's a 1,000-dimensional space. There could be many interesting directions that you want to explore, right? Okay. Sorry. Any other questions? Yeah. Okay, so let's see. Let's see if I got this right. So this is a synapse from input I to output J. From input J to output I, and then it depends on the sum of the same inputs, but going to different outputs. Okay, so this synapse has to know the weight of those synapses multiplied by the activities of all other neurons. Is that okay? Yeah, sorry, this with the transposed, it's tricky. Okay? Why not use? Right. 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 So the idea is that so think of this as like visual inputs, right? You have 1 million axons in your optic nerve. So it's a 1 million dimensional space. And these are like, you can think of them as 1 million pixels. But do you really want to operate in your representation perceptual space in this 1 million dimensional space? No. You want to simplify it to things that you can really manipulate with cognitively. Like these are the people you know, these are the objects you can use, and these are features that form a lower dimensional space than million. I don't know, it may be not, what I'm saying it's not 1, but it's not 1 million either. It may be a 10 or 20, but it's in this intermediate range. And so out of 1 million, we want to pull out those 10 or 20 principal directions. That's the idea. So we don't get swamped by the 1 million dimensional space, but we pull out the 10 or 20 the features that we care about. Does that help? Yes. Okay, that's a good, so that's a good question. So let's imagine that our task indeed is to classify visual inputs. Let's say, pictures with cats and dogs, right? So you want to learn a classifier. The question is, can you learn this classifier directly on the pixel level representation? And that's going to be very, very hard, right? So what actually you have to do is to transform this representation to some higher order representation through some nonlinear transformations where you can learn a linear classifier that will differentiate cats and dogs. So that's what deep learning does. That's what you need all those layers for that perform this transformation. So it's very hard to actually do this classification task in the original pixel space. And that's why we're doing this. Does that help? Okay. And generally, so I was going to show this in another slide, but generally it's believed that natural images, even though they're million-dimensional, million-dimensional space nominally because they're million pixels, that space is not uniformly spanned like you would if you randomly generated a million-dimensional vector. But there is a low-dimensional manifold in that million-dimensional space on which natural images can live. So, for example, if you were to generate a random million-dimensional image, just a random matrix, and say, okay, this is my pixel intensities, it will look like white noise. It will not look like a real image. And that's because most of the random vectors in that million-dimensional space, they don't live on that low-dimensional manifold. But the natural images do. And so our visual system is very good in pulling out that lower dimensional manifold. And that's what we're trying to model here. Does that, okay. All right. So, yeah, so we ran into a problem. So the Fourier rule, which worked great for a single neuron, doesn't really work for multiple components, for multiple neurons. And this learning rule is just part of a problem, right? This is already non-local. But what about this term? What about the lateral connections? Okay, so when we run this algorithm on the computer, we can compute W and then compute the Gramian. But the synapses, how are they going to do this? How are they going to compute this crazy products, each of which is updated according to this rule? This is just like completely infeasible biologically. And so that's why this approach ran into a problem. And there are two ways out of this. Right? So if you want to pursue online dimensionality reduction for more than a single component, there are two things you could do. One is to use just the learning rules we derived, which are not biologically plausible because they're non-local, and you just use them for kind of machine learning tasks, but you cannot use them to model the brain. Okay? Or if you want to model the brain, what people did is they said, okay, we have to come up with the learning rules that actually work, are local and biologically plausible in nature, so let's guess them. Let's just write down a learning rule that is local and that kind of works. That's also possible, but then you don't really know what that learning rule achieves. Will it always compute principal components for every data set or not? You don't know. Usually it's hard to prove if you didn't derive those rules, and you just can numerically simulate, and then you don't know if it will always work. So that was kind of the state of the art for a long time, and there was no biologically plausible principled approach to PCA. And when I say that, there is a caveat. This is all true if you wanted to do this operation in a single layer. If you're allowed to have two layers of neurons, then you can actually do it, and there is a network that would do it. But when we try to map this network onto biological examples, like early sensory systems, that actually wasn't a good match. So we decided that that wasn't a good approach. So at this point, let's see, how much more time do I have? Okay. Okay. Well, yeah, okay. So I have, so there is a new part to the talk. So should I just go on? Okay. All right. So in the normative approach, you know, starting with the optimization problem, derived an online algorithm, mapping onto the neural network, we ran into a problem. Okay. So what are we going to do? Well, the bigness of the normative approach is that we had to guess the objective function. But it's also a strength. We can take another guess, right? So we can go back and try to find another objective function and try to follow through with this program and see if we can get principled but also biologically plausible local learning rules. And that's basically what we did. And because there was this failure for multiple neurons, we came up with what is called the similarity matching objective. And that objective, oh, thank you. That objective will allow us to overcome this problem. So what is similarity, and why are we using it? So similarity, the way we define it, is just the inner product between the pairs of inputs, and that's this input similarity, and the inner product between the pairs of outputs. That's the output similarity. That's it. It's just like the cosine similarity, except that it has the magnitude too. The basic idea then is, instead of focusing on the data vectors themselves, we focus on the similarities between those data vectors. So for example, if we have 3 data vectors, green, blue, and red, in the input space, then which vector is blue most similar to? Red. So blue and red are most similar, and green is less similar. So imagine that we were to require that the same relationship is preserved in the output space. We don't require anything about the reconstruction. There is no reconstruction step. We are not saying that I need to use this blue vector to know where that blue vector is. All I'm saying is that I preserve the similarity of the inputs in the similarity of the outputs. And then, of course, even the dimension of the output space can be different from the dimension of the input space. For example, I could have a 1,000,000-dimensional space here and a 10-dimensional space here, and I want to try to preserve similarity. How do I do that? Well, one way to do that is, for example, okay, so this is, by the way, these are the similarity matrices. So this is a matrix of pairwise similarity. So this similarity of the data point in the row. So first data point with the data point in the column. Okay, so that's the matrix of all the similarities of the inputs. And that's the matrix of all the similarities of the outputs. We want them to be as similar as possible. And we can perhaps achieve that by minimizing the squared error of the element-wise error between those two similarity matrices. So instead of the reconstruction error, when we try to minimize the difference between the actual x and the reconstructed x, we minimize the difference between the similarity of inputs and the similarity of outputs. Okay. All right. K is the dimension of the output space. So like in a single neuron, there will be one and other. Yes. Yes. So basically, the well-kept secret is that the optimum of this similarity is also PCA. Or I should be more careful, it's a projection to the principal subspace, okay, which is up to an orthogonal rotation is the same as PCA. So that's the solution of the similarity. Even though it looks very different from reconstruction objective, it actually has the same solution. And that was known in the field of multidimensional scaling, which does exactly what you described. So you can use the same logic as in PCA. You can look for an elbow and explain covariance as the number of components, and then choose the point where that elbow happens as the number of your components. Okay? So basically, this is just crazy, and I didn't know, actually. I came to this objective in my own way through just reverse engineering Hebbian rules and trying to see how I can get an objective that will give me Hebbian rules. And I didn't know that this exists and this gives PCA, but once I wrote it down, it turned out that that has already been proven. And so the idea is that we're going to use this objective instead of the reconstruction objective to derive the neuronal network. Any questions at this point? Okay. So, well, I have a question for you. Is this really does this a good object for deriving, a neuronal online algorithm? Well, it doesn't look like a good objective. Can you tell why? That's certainly true. It's still a linear objective. But what I'm saying here is that it seems a little bit difficult to implement in the online setting, right? I'm sorry? Why tau? Right. So these are outputs at different time points, t and tau. And they're all entangled with the inputs at different time points. So it seems that to compute this objective function, you need the whole data set at once. And this is not something a neuron can do because the neuron exists in an online streaming method where at each time point it gets xt and it needs to compute a corresponding yt. And it cannot remember very much. It doesn't have a big RAM to store all the past data set in. So it has to just store the very minimum and somehow compute this. But how can it do? Because here all the data seems to be entangled together and between the inputs and outputs. So how is this objective function any good as a model of a neuron in terms of online algorithm? That means that it's the same data point, right? So here I sum over all data points from 1 to capital T. So it's like this is T and this is tau. So I have capital T data points, okay? And I need to compute similarity, pairwise similarity between all of the pairs. So I call this index T and this index tau. And then this shows me all these terms here. Okay? Good? Okay. So, but the problem is that then I have to combine, say, x1 with the x capital T to optimize this objective. So what the neuron is going to put x1 in memory, and when xt comes in, it will use it. And then the same should be true with all the other x's, right? And the neuron doesn't have the memory to store all the past data. And if we could do that, then as someone suggested, why do the dimensionality reduction? Just keep all the data. Okay, so that's a problem, that this objective didn't seem like a good choice for an online algorithm. And so what we showed, that actually there is a mathematical trick that makes this objective very amenable to an online algorithm. And to see this, what we have to do is to just expand the square, and then we'll have 3 terms. The quartic x term does not depend on y, so we can ignore it. And then there is a term which has 2 y's and 2 x's. That's the term. And you see all the t's and tau's are entangled here. But with algebra, what we can do, we can move x tau's around, right? Because we can flip this inner product like this, and it's still the same thing. And then y tau and x tau are together. And then we can bring the sum inside, and that sum is just a sample covariance between y and x. And over a long time, when t is sufficiently big, the covariance doesn't change much from time point to time point. And so we can just use wyx, which is the estimate of covariance from the previous time step. like we use the weights in the Euler rule. And then we can compute yt by this iteration, which you can get by just simply using this term and this quartic term with the same trick, which can be done with the same kind of architecture that we had before for the multi-neuron Euler rule. Okay? But the learning rules become completely local because there was the sum here that now disappeared. Why? Because we set up an objective where we got this covariance, which is a matrix, the components of which are encoded in the feed-forward synaptic weights for yx covariance, and yy is from the quartic y term is encoded here. And both of those learning rule updates are completely local. Okay? And you can also check that for a single neuron, this becomes the familiar Euler rule, right? So that, you know, if you just have one neuron, it's exactly the same thing as before because it's just the top principal component. So this is a generalization of the Euler rule to multiple channels, to multiple neurons, which preserves the locality of the learning rules. And therefore is biologically plausible. Yeah. Yes, yes. So, okay, so that's very perceptive. So there are actually different versions of this algorithm. In the one I use, the reason you have this sum is because you have a term here in the quartic y where it's you know, it's a term where this index of y is the same, yt, okay? And so to compute yt here, you have to divide by that sum, right? So it's this sum that is in here, okay? And so you divide by this to compute yt. Now the question is, how can you implement it online? Well, you don't have to remember all y's for that. All you have to do is to keep one number that is a running sum of y and update at each step by adding a new y squared. Okay? So this adds one more, additional scalar number per neuron. And that's not a big deal, right? So we stored all the elements of the covariance in the synapses for feed-forward weights and the output covariance in the lateral connection weights. And this way we don't have to store individual data points. So this is the way to do the dimensionality reduction now, where we just store the sufficient statistic, which is just the input-output covariance and output-output covariance. And the beauty, again, is that the learning rules are completely local and therefore can be implemented biologically. Yes. So offline you can prove, so if you take the subjective function, you take an SVD, a single value decomposition of x and y, and then you can show that what you need to do to minimize it is to align y with the top singular vectors of x, which is the eigenvectors of x transpose x. Okay? So that's how you prove it, just through taking SVD. And this is not very widely known, but in the multidimensional scaling field, that's a very old result. Now, the next question may be, okay, you know that offline, this will give you PCA. But if you run this algorithm online on the streaming data, how do you know it will be PCA? So these things are usually very, very hard to prove. And in this case, I don't know actually if the rigorous proof exists. In practice, it always works. But I'm not actually completely aware if someone has proven in the online setting that this will still give you PCA. But, you know, practically it's true, but mathematically. No, anytime. Yes. Yeah. Yeah, that's an excellent question. So okay, I don't know if I should go back, but the feedback synaptic weight vector is very similar to feed forward. It's not exactly the same, but it's very similar. And because, you know, synaptic weight only known approximately, I think that's okay. So we can probably capture the top principal component. But the other vectors of other inhibitory neurons, I don't think we can get it with PCA because this is a linear, you know, our model has linear neurons. And the real neurons are rectified. So when we model it with rectified neurons, we have a slightly better fit. So I think that's what's missing there. Yeah. Uh-huh. Right. Okay. So let me answer the second question. Yes. So the best experiment would be if we could measure the activity of pre and post synaptic neurons, compute the prediction synaptic weights from PCA, and then measure them physiologically or anatomically. So it's difficult experiment, but given today's technological achievements, I think it can be done. So, but I don't think, I mean, there are some simpler situations where, you know, we know that we can influence the synaptic weights, that they respond to activity, but it hasn't been tested exhaustively with, you know, all components. And then the other question was, when you said one neuron contributes to the covariance the most, did you mean the presynaptic neuron? Yeah, then it would have to, yes, the synaptic plasticity would have to adapt. Yes. 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 Okay. So I think this might be, oh, there is an, okay, sorry. Say it again. Uh-huh. Yeah, so this could be some kind of a activity sensitive mechanism like the calcium, some sort of kinase or something like that, right? So there are a lot of molecules, a lot of proteins in the neuron, and you only need one number to store one number here, right? So I think that's okay. All right. So this is basically where we are. So we started to, to summarize this. We started with this normative approach using a reconstruction objective, and it kind of worked for single neuron, and that's what Oya did. and it failed for multiple neurons because, we couldn't have both principled and biological plausible learning rules. And then this new approach, the similarity matching objective, gives you a different mathematical objective that needs to be optimized. But in this case, it has the same solution, also PCA. But because of the properties, mathematical properties of the similarity matching objective, when we derive learning rules from it, they turn out to be local. And we've done this for linear neurons, but of course the real neurons are not quite linear. And so the next two parts are about making this even more biologically realistic by adding rectification and the two compartment neurons with calcium plateaus. So I think that will have to be next time. Correct? Okay, thank you.